Okay, so now we are within our diagram packet, and you guys should be opened up to, I want to say it's like the 5th page, the front of the 5th page within that diagram packet. And let's start out in a regular view rather than landscape. So I'm looking at the page that talks about transcription of a gene. So we're going to look at two processes, transcription as well as translation. Transcription is that first step of protein synthesis. The second step is going to be translation. So the first thing, we got to transcribe the gene. We need to put that language of my DNA code into a language that messenger RNA can understand. So we're kind of transcribing it. Once we send it out to the cytoplasm, that's where we're going to translate it into a protein, put amino acids together. Since we are using DNA and we're going to make it into messenger RNA, we know that has to take place in the nucleus because we talked about anything involving DNA has to take place in the nucleus. So once again, transcription inside the nucleus, first step of protein synthesis. Spin that page around. Let's look at it landscape view. Alrighty. So here's my DNA strand. Let's pretend this box is my nucleus. And here's my DNA strand. And we're only looking at a segment of it because we're only going to be transcribing a gene, a segment of DNA that's going to code for a protein or a trait. So this part stays all wound up. The rest of my chromosome will still be wound up. I'm only looking at this short strand. So the yellow is my original DNA double strand. And it kind of starts out the same way. So helicase is still going to come in and do its job just like it did for replication. It's going to unwind it. It's going to break those hydrogen bonds. Instead of using DNA polymerase, however, now we're going to use RNA polymerase. And RNA polymerase is doing the same job. It's just going to go through and it's going to go and make RNA nucleotides now. So if we think about RNA nucleotides, we're still dealing with a sugar, a phosphate, and a nitrogen base. My sugar is going to be ribose, however. My nitrogen base, I have uracil replacing thymine, and this RNA is going to be single strand. So my nucleotide is very, very similar. What I'm doing here is I'm just taking this strand of my original DNA, and it's the same strand. I'm just moving it over here so it's easy for us to work with. So we're just kind of pretending we're shifting this over. You're going to notice that every 3 base pairs on DNA, once again, is called a triplet. So I can say I have 1, 2, 3 triplets along my DNA strand. I have TGC, my first triplet, AGT second, TCA is my third triplet. DNA, just like when I replicated it, is going to get read 3' to 5'. My messenger RNA that I'm making off of this template strand gets made 5' to 3. The same way it did for replication. So that part does not change. So now my RNA polymerase, because I'm making RNA nucleotides joined together, is going to attach to my DNA strand. And it's going to start reading those base pairs. And he's going to go along and go, okay, from my 3' to my 5' end, there's a thymine. Let me add an adenine. I still have adenine in RNA. Here's a guanine. I'll attach a cytosine. Here's a cytosine. Let me attach guanine. Here's an adenine. Ooh, I can't put thymine there, but uracil will replace it. And I'll keep going through. You're going to notice every 3 base pairs in messenger RNA are known as codons. So my codons are complementary to my triplets. A goes with T, G goes with C, C goes with G. They are complementary base pairs together. It's just I'm replacing uracil. I'm replacing thymine with uracil. So if I have 3 triplets here, I'm going to have 3 codons here. The DNA, once again, is getting read 3 to 5' by the RNA polymerase. My new messenger RNA strand is being made 5 to 3. It's also anti-parallel. So once this is done, this pink strand is all joined together. It's coded for. I've kind of hit that start codon or the start area. I've hit a stop area. Said, hey, this is where the entire code that you're going to need in order to make the protein that we need. This messenger RNA strand, single-stranded molecule, is going to break off from DNA, and it's going to exit a pore within the nucleus. At this point, RNA is out in cytoplasm. My original DNA, its job is done, so it's just going to come back together again, my original strands, in that double helix. If we look at it, just one more little view on that, you guys on the next page should see this particular structure, and it's basically looking at the same thing. It's showing you what that RNA nucleotide looks like, same thing that we saw within our DNA picture that we have. Remember that all of your RNAs are kind of made within the nucleus, so here's my little transfer RNA. He's going to be a little taxi cat. On the bottom of my transfer RNA, you'll see this on the next diagram as well, I'm going to have 3 base pairs, just like my other RNAs, messenger RNA specifically. These 3 base pairs are known as an anticodon. Once transfer RNA is made, once ribosomal RNA is made, it's going to leave the nucleus and do its job out in the cytoplasm. Within your messenger RNA, we talked about these codons or 3 base pairs. There is a start codon. It is AUG. So when you see the base pair is AUG, you know that's going to be the first codon making up that particular protein. When my ribosome eventually is going to read either one of these or these 3 base pairs, UAA, UAG, UGA, or 3 codons, that's when it knows to stop making that particular protein. They are called stop codons. So these will always be found in messenger RNA that you see here. Once again, DNA gets read 3' to 5'. My messenger RNA is being made 5 to 3, and you guys can see them being added here. This blue, we had in the last diagram as your DNA polymerase. This is now my RNA polymerase. So once those strands are done, messenger RNA breaks off, the original strands will come right back together again. I'm only opening up the section that I need. Messenger RNA leaves, it goes out into the cytoplasm. Once out in the cytoplasm, you guys are going to see things like this. So once again, let's go regular view on this so we can look to see what we're doing. This entire page that you have here of protein synthesis is now called translation. It's my second step. Here we're dealing with messenger RNA, and it's going to be made into a protein. I'm going to utilize rRNA, I'm going to utilize tRNA, as well as messenger RNA. This is all taking place in the cytoplasm. This is where proteins are finally put together. Let's flip it back to landscape view. All right, so my pink strand was my messenger RNA. The little blue structure you have here is my ribosome. This ribosome, this blue structure, 2 units, a big subunit, a small subunit, is joined together by rRNA. Kind of glues them together and activates them. It's kind of like a little decoder ring. My transfer RNAs are these guys. They kind of are shaped like little T's. Think like taxis. So these taxi calves or transfer RNAs have 3 base pairs on the bottom of them. We said that they are known as anticodons. Their anticodon tells them which specific amino acid can be their passenger, which one they're picking up. So they just can't go out and go, oh, let me just pick up valine, because that's who he feels like picking up. His code's going to tell him specifically which ones to pick up and bring into my messenger RNA strand. So they're all floating around the cytoplasm. Those amino acids are coming from foods that you eat. So let's go back to my pink strand that you guys see here. This pink strand is my messenger RNA. Notice I've already split them up into codons, every 3 base pairs. This messenger RNA strand is going to enter into my ribosome in this direction. So he's going to get read, and it's kind of different than what we've seen so far. He will enter in 5' end in first. So he will actually get read 5' to 3'. So my ribosome is moving down my messenger RNA strand in this fashion. So my ribosome started way over here at AUG. He could have started millions of base pairs before then, but until he reads that particular code, absolutely nothing is going to happen. If you remember that, AUG is what is known as my start codon. As soon as the ribosome reads that, he goes, ooh, this is going to be my first amino acid that I'm going to put in to make a particular protein. So he's actually going to call in a particular transfer RNA. He's kind of already left, so I drew one in over here. This little transfer RNA is going to come in, hook up. A goes with U goes with A, G goes with C. Oh, my anticodon on transfer RNA is complementary to my messenger RNA codon. And he brings in his amino acid. Then my ribosome shifts down. And he hooks up to the next one. He brings in his amino acid. These 2 amino acids are now going to bond together through a peptide bond. Dehydration synthesis we saw on the front page of our notes. Once we have 2 amino acids joined together, it is known as a dipeptide. 2 of them joined together. The ribosome slides down, so let's say we're on CGA. He yells out, brings in the particular transfer RNA. C goes with G, G goes with C, A goes with U. He brings in his amino acid. Dehydration synthesis. Now I'm growing that polypeptide chain. This step is called elongation, step number 2 here. I'm adding amino acids to that particular protein. Ribosome shifts down. Now I'm going to look at ACA. He's yelling out. He goes, oh, bring in UGU as my transfer RNA. A goes with U, C goes with G, A goes with U. Hooks my amino acid. He's going to shift, and the next one comes down, and it's going to keep going and keep going and keep going. I'm going to keep adding amino acids. My smallest protein is 50 amino acids. The longest one could be thousands and thousands and thousands of them. So eventually, as I keep going all the way down and keep adding all of my amino acids there, my ribosome is shifting down. Eventually, I'm going to reach this code here. Oh, it says UAA. UAA is one of my 3 stop codons. So I have UAA, UAG, UGA, and those are in the note sheets that we went through earlier. If I read any one of those 3 stop codons, there is no transfer RNA that will hook to that. So there are no transfer RNAs that are going to read AUU. It doesn't happen. That's why we only have 30 or 61 transfer RNAs and 64 different codons in messenger RNA. Okay, so if we flip to the next page on that, you'll see those processes one more time. And it's easier if you guys kind of separate these phases out. Here's initiation, here's elongation, here's termination, and you guys can see that a little bit better. It shows you the differences between codons and anticodons. Codons in messenger RNA, anticodons in transfer RNA. First step, this is all out in the cytoplasm because it's translation. The little dots here are my ribosomes, so what's going to happen as messenger RNA enters the cytoplasm, the 2 subunits of ribosome are going to be joined by ribosomal RNA or rRNA and activated. My messenger RNA enters the ribosome 5' end first, and the ribosome reads that start codon AUG where he starts to call in his appropriate transfer RNA with my amino acids on the back side of it. That step is initiation, so it's reading the start codon. The second step is elongation. I'm growing it. So there's an enzyme as we're coming through called peptidyl transferase. This is what's going to release the transfer RNA from my amino acid because only 2 tRNAs can stay on the ribosome at a time. So as that ribosome shifts down, he's going to get kicked off. This blue one's going to get kicked off, but he leaves behind his amino acid. And the reason is because it joined to the next amino acid by a peptide bond. Dehydration synthesis. We took out water. The bond is strong there. I can break off that transfer RNA, and that's what peptidyl transferase does. It releases that transfer RNA like we see here from my amino acid, and it allows another one to come in. Remember, only 2 transfer RNAs can fit on that particular ribosome at a time. He comes in, codon UUU matches with anticodon AAA. They briefly bond together. It allows my amino acids to peptide bond. It moves down, peptidyl transferase kicks off this one, and we have elongation. We're growing that polypeptide chain. Now we have termination, last step. So with this termination, we are growing the polypeptide chain. Remember, 2 of them is called a dipeptide. A whole bunch of them is a polypeptide or a protein. Eventually, we reach one of 3 stop codons. Here, it's going to be UGA. It could also be UAA or UAG. So there's the 3 that you have there. There is no transfer RNA for that particular codon. It just says, okay, that's stopping. Let's end it. My amino acids break off, which is now known as a protein. My ribosome can either break down or he'll find another messenger RNA strand. And my messenger RNA can get recycled. It can make more protein, or it can kind of get broken down along the way. The next step that we'll see a little bit more probably tomorrow, we're going to see, now what happens to that protein? We have this protein that's kind of floating around within our cytoplasm. Maybe it's an enzyme. Where do we need it? Where does it go? How do we get it there? And we're going to utilize other cell parts for that. Later today, just like you guys saw with the replication hands-on DNA, you guys will see a video going through protein synthesis hands-on. I encourage you, if you have questions, post stuff on Google Classroom that other classmates are able to see so you guys can kind of answer questions back and forth. I can answer questions so that everybody else can see your questions as well. It may help others understand. But please, please, please make sure that you guys are going through these notes, watching the videos, going back and reviewing them. There will be some tests online that will be their assessments for this as we work our way through it. Okay, let me know if you have any questions. Thanks.